Good morning, dear students, and welcome back in our video lecture with the same topic, cell. So today we are going to study next cell organelle, which is called Golgi apparatus. Also, it is called Golgi bodies. So, my first question is, who discovered these Golgi bodies? You have any idea about this? Okay, I will tell you. Golgi bodies was first observed by Italian doctor Camillo Golgi. He had also established a cell staining technique called black reaction using silver nitrate solution, but that is not for our syllabus. What we are going to study about only discovery of Golgi body. So let's start with the discovery of Golgi body. Camillo Golgi was the scientist who just studied the structure of central nervous system by observing the brains of owl and cat. He found that in the nerve cells of cat, Golgi bodies were present, and he just called them internal reticular apparatus. He thought that these Golgi bodies only present in nerve cells and played major role in the functioning of nervous system. But after that, he found that Golgi bodies are present in all eukaryotic cells. So, what is mean by Golgi body? Golgi body is the membrane bound organelle. So you can see here in the diagram. Normally it is found in all eukaryotic cells. Exceptional is RBCs, sieve tubes in plants, male gametes of bryophytes and pteridophytes and prokaryotic cells. These are the exceptional case. Otherwise, these Golgi bodies, this Golgi apparatus is found in all types of eukaryotic cells. So, what is the structure of Golgi apparatus? You can see in the diagram, it consists of stacks of flattened membrane sacs. These all sacs are just arranged parallel to each other, and these sacs are called cisternae. Cisternae are placed one above the another in parallel manner. These membranes often have connections with the membrane of endoplasmic reticulum and located near the nucleus. Means normally whenever you are observing any cell, you found that Golgi apparatus are normally present near to the nucleus, and they are just connected to endoplasmic reticulum. Then this Golgi apparatus consists of two phases, cis phase and trans phase. The phase, the part of Golgi body is near to the nucleus, facing to nucleus is cis phase, and outer side part is called trans phase. So cis phase plays role receiving material from endoplasmic reticulum, and trans phase plays role in delivering the same to other cellular parts. Then when we are talking about its function, what is the function of Golgi body? I will explain you by giving another example. In each and every factory, different packaging material is there. Different modification, packaging, and storing material is department is there. What will happen inside that department? So whatever the raw material which get manufactured into that factory, that raw material just modified, then stored, modified and packed into that department. That is the work of that factory or that packaging department. So same thing happen here in Golgi apparatus. Proteins, lipids, they are produced by endoplasmic reticulum. We have discussed about this. Proteins are synthesized in endoplasmic reticulum because endoplasmic reticulum consists of ribosomes, and the ribosomes are responsible for synthesis of protein. So whatever proteins, whatever lipids, the material, raw material get produced into the cell, that raw material just come to cis phase of that Golgi apparatus, and then from cis phase this material just passes to trans phase. And you can see here there are many membranes, many just sacs are arranged in parallel manner, and when that raw material comes in each sac, the modification storing takes place. That material, raw material gets modified, and then just several reactions takes place inside that sacs, and that reactions leads to modification of that raw material, and as it passes to next level, next sac, it get modified, and after completion of modification they are just packed into vesicle, and then this modified vesicles, this modified material, they just pass to the different parts of cell where it is necessary, or they just reach to destination. Got it, student? What is the function of Golgi apparatus in a cell? It behaves like department modification department in factory. What happens in modification department of factory? All raw material, it gets stored, it get modified, it get packed into that department and then sent to their destination. Like this, in Golgi apparatus also, whatever proteins, lipids, whatever form, that raw material just come to cis phase, then from cis phase it just pass to different sacs of that Golgi apparatus, and each and every sac it get modified, and then after completion of modification, this raw material get packed into the vesicle, and then it comes out of that transphase and just pass to the destination, or just pass to the different parts of cell where it is necessary. So we will study detail what is the function of Golgi apparatus. Golgi apparatus is secretory organelle of the cell. The proteins, lipids, steroids and some other substances pass from endoplasmic reticulum to the Golgi apparatus. The Golgi apparatus modifies and packages these biochemicals into tiny secretory vesicles. So vesicles are nothing but small, you can see in the diagram, small spherical structures are there. Small circular structures are there near to Golgi apparatus. These small structures are nothing but vesicles. These secretory vesicles then pass out from Golgi apparatus to various parts of cell or outside the cell also through exocytosis. Then what is exocytosis? Exocytosis is the process that is used to transport material from inside the cell to external part of the cell by using energy. So examples: mucus, sweat, proteins, saliva, tears, pigments, enzymes, hormones, and in plant cells, gums and wax. These are the examples. Means these are the materials which get modified into this Golgi apparatus and then sent to destination. It helps in the storage, modification, and packaging of secretory products. Whatever product gets secreted into the cell, it gets stored, modified and packed into this Golgi apparatus. It helps in the formation of cell plate, cell wall, plasma membrane and lysosomes during cell division. We have studied about cell division. What is cell division? Cell division is nothing but conversion of one cell into two cell, 2 to 4. So during cell division, this Golgi apparatus just form, or it helps in the formation of plasma membrane, cell wall, cell plate and lysosomes also. What is lysosome? That we will study in our next video lecture. It is involved in the modification and maturation of acrosome also. This is a particular function of Golgi apparatus. It involved in the modification and maturation of acrosome. What is acrosome? Acrosome is special structure at the tip of sperm, and its modification and maturation in this is also involved in the functioning of Golgi apparatus. So complex sugar can be made from simple sugars in Golgi apparatus in some cases. So what happened? Some proteins or some simple sugars just reaches to cis phase of that Golgi apparatus, and as this simple sugar passes from these sacs, from one sac to next sac, from next sac to another sac, then what happens inside that Golgi apparatus? Many chemical reactions takes place, many biochemical activities done, and this simple sugar get converted into complex sugar. Then it is packed into vesicle and it comes out from transphase, and then it reaches to destination. It reaches to the desired that particular part of cell where it is required. So Golgi apparatus is nothing but it behaves like modification department of any company, any factory, and these are the functions of Golgi apparatus. So I hope you got all points related to Golgi apparatus. Again, we will revise all points. Golgi apparatus is a membrane-bound organelle. It is found in all eukaryotic cells. Normally, when you observe any cell, you just found it is near to nucleus, but not all time. The protein synthesis is done by ribosomes. Normally, we have studied about the synthesis of protein, how it is done by ribosomes, and the proteins, lipids, whatever is secreted into the cell, it behaves like raw material for Golgi apparatus. All this just come into cis phase of Golgi apparatus. Then from cis phase, it is come to the internal structure, sac-like structure, and as it is passes from one sac to next sac, several modifications takes place into that raw material. As it passes from one sac to next sac, that raw material get modified, and after completion of modification, they just pack into small vesicles, and then they just come out of that transphase and reaches to destination. Okay, so what is the function of Golgi apparatus? First, we will again revise structure. It consisting of stacks of flattened membrane sacs called cisternae. Means Golgi apparatus consists of cisternae and vesicles. So cisternae are placed one above the another in parallel manner, and these membranes are often have connection with membranes of endoplasmic reticulum. They are normally located to near to nucleus. And these two distinct phases of Golgi apparatus are cis phase and trans phase. Cis phase is facing towards nucleus, means the part of Golgi apparatus which is near to nucleus is cis phase. Then cis phase plays important role in receiving material from endoplasmic reticulum, and transphase plays important role in delivering the same to other cellular part. Then what are the functions? This Golgi apparatus is secretory organelle of the cell. Proteins, lipids, steroids, and some other substances pass from ER to the Golgi apparatus. Then they are modified, packed, and during the biochemical reactions, and they are just packed into some tiny vesicles. These vesicles then pass out from Golgi apparatus to various part of cell or outside the cell also. It helps in the storage, modification, and packaging of secretory products. It helps in the formation of cell plate, cell wall, plasma membrane, and lysosomes during cell division. Also, it involves in the modification and maturation of acrosome. What is acrosome? Acrosome is special structure at the tip of sperm. And complex sugar can be made from simple sugar, or you can say simple sugar can be converted into complex sugar in Golgi apparatus in some cases. So I hope you got all points related to Golgi apparatus. What is meant by Golgi apparatus? What is the structure of Golgi apparatus, and what is the function? What are the functions of Golgi apparatus? Students, if you have any doubt, you can ask me. Thank you.